really a novel treatment for protoporphyrias, both EPP and XLP. This has a few parts, and I'll go through it fairly quickly. The key takeaways from these beacon data, which are data that have come from a study done in Australia, are as follows. A dose-dependent reduction in protoporphyrin 9 levels, PP9, more than 30%, both at low and higher doses. The two doses used were 20 milligrams once a day and 60 milligrams once a day. There turned out to be significant effects to improve sunlight tolerance, decrease phototoxic reactions, perceived benefit in terms of overall quality of life, and of particular interest is that the drug was remarkably well tolerated, particularly with no real changes of significance in levels of blood hemoglobin. I think most of you know that EPP is this relatively rare condition which can be debilitating. People have to learn to really stay out of the sun. If they get in the sun, they can get quite ill from swelling, pain, tingling, and it can last for days, even up to several days. And there's also the phenomenon of sensitization. So being exposed one day can lead to greater sensitivity on the next day and subsequent days. It can be debilitating, occasionally even life-threatening, because there is a rare occurrence of what's called protoporphyric hepatopathy, or a liver disease related to the excess of protoporphyrin. And currently there is no real cure other than bone marrow transplantation. We have transplanted some livers of people that had liver failure, but that in itself is not a cure. It obviously is life-saving when these people have advanced liver disease, but the underlying disorder of overproduction of protoporphyrin by the developing red cells continues. On the skin, the effect of protoporphyrin 9, which is shown in the center of the slide there. So this is a tetrapyrrole composed of these 4 pyrrole rings with various side chains: methyl, vinyl, methylpropionate, propionate, methyl, as one goes from the top left around to the bottom right. And in the skin, the porphyrin ring absorbs light, particularly around the so-called Thoré band, 410 nanometers, which is in the blue end of the spectrum. And that in turn then gives up energy and heat. It causes oxidative and other damage, pro-inflammatory damage to the skin, and produces the symptoms. The accumulation in the liver can lead to bile plugs that are rich in protoporphyrin, often to gallstone formation. About 25% of people eventually need to have their gallbladders out because of symptomatic gallstones, which are rich in protoporphyrin, and rarely end-stage liver disease, as I've said. There may be some other complications, such as vitamin D deficiency, because people are staying away from the sun. And of course, there are definitely effects on social functioning and social life, for sure. Now, Bitopertin, I hope you can see my pointer, is a small compound that was originally developed for other possible reasons. It is an inhibitor of a transporter of glycine. Glycine is an amino acid, as many of you know. and it is an essential substrate for the first and normally rate controlling step in overall heme synthesis, namely the condensation of this molecule of glycine with succinyl-CoA to form the first committed intermediate of the pathway delta aminolevulinic acid, ALA. That's carried out by the enzyme ALA synthase. Then through a series of steps, the ALA is eventually converted to protoporphyrin, and in the final step of the pathway for heme synthesis, a molecule of ferrous iron is inserted into the protoporphyrin ring to form heme, and that heme is used for a variety of things in the body, but quantitatively, the greatest user of heme every day is hemoglobin, the thing that makes our blood red and carries oxygen and carbon dioxide. The common mutation in protoporphyria is in ferrochelatase, this final step of the pathway, but there turns out to be another disorder that phenotypically looks the same, but is due to a gain of function mutation of this first and normally rate controlling step of ALA synthase. And that goes by the term X-linked protoporphyria, because the ALA synthase 2, the enzyme that is carrying out this first step of the pathway in all of us, is found on the X chromosome. That's why it's called X-linked protoporphyria. It's been found that 30% reduction or more in proto levels has a significant effect, a favorable effect on photosensitivity. And this has been known for years from observations that pregnant women experience a fairly marked reduction in proto levels and a fairly marked reduction in the severity of their photosensitivity. And another more recent study was a protoporphyrin inactivation study in which the blood from patients with EPP was exposed to light outside their body and then returned to the patient. This procedure reduced proto levels by about 30% and was associated with a marked improvement in tolerance to sunlight. This was a recent paper from Denmark in the journal Photodiagnosis and Photodynamic Therapy. In preclinical studies of mouse models of EPP and XLP, as you can see from this slide, there was a significant fall in proto levels. This was a controlled diet. This was mice with a defect in ferrochelatase, fed Bitopertin, 100 parts per million in the diet. And as you can see, there was a significant decrease, 45% decrease in proto levels. This was a different model. This was the gain of function mutation in ALA synthase, a 73% reduction in proto levels. And of significance also was that liver fibrosis, which occurs in these mice fed the control diet, was markedly improved in a dose-dependent way in the presence of Bitopertin. So very exciting results preclinically. And that was one of the stimuli for going on to the first clinical studies, so-called Beacon study. This involved a total of 22 subjects, all in Australia. They all received Bitopertin at different doses. And there were 8 who were enrolled to receive 20 milligrams of bitopertin per day, 7 to receive 60 milligrams of bitopertin per day. As you can see, the majority completed. I mean, the study is actually still ongoing, so that so far, or at the time of the data cutoff for these slides, only one had completed the entire 169 days. The others were still in the study. And the trial endpoints were changes in Proto-9 levels, light tolerance, time to prodromal symptom, and overall safety and tolerability. And these are the main results of proto levels. The blue shows the lower dose, 20 milligrams per day, the orange, the higher dose. Notice that by day 29, there definitely was a significant decrease in whole blood protoporphyrin levels, and that it was sustained throughout the rest of the time. Admittedly, there's only one subject here at day 169 at the time of this cutoff, so we want additional data, of course, to support this. So these are the individual patients. As you can see, the one that finished the entire 169 days was being treated with 60 milligrams per day. And in general, those treated with higher doses had a greater effect, but all of them had a fall except for just this one patient, and this was only at the earliest time, day 15. In terms of light tolerance, these are the results. So at baseline, this particular subject could only spend less than 5 minutes before having this prodromal symptom develop. No significant improvements at day 20 or 27. By day 41, a bit of an increase. By day 63, as you can see, more of an increase. And then by day 88, a very marked increase in ability to be out in the sun. Day 96, it was lower. So if you look at the entire thing, it probably went up to around 200 minutes from about 5 minutes. And there was about a 30% decrease in proto levels. The baseline proto was pretty high in this particular subject at 2740 micrograms per deciliter of whole blood. Here's another patient, again, very short time at baseline. By day 29, some improvement. Then again, not much tolerance, but at day 74, 255 minutes, and at subsequent days, again, quite a marked improvement in the ability to be out in the sun, with overall a decrease in proto levels that was as high as 72% at this higher dose, 60 milligrams given once a day. In terms of days without symptoms, there was a marked improvement, and prodrome-free sunlight challenges went from 0 to 50% of patients during the course of the study. The average time to prodrome, as you can see, went up gradually, and the weekly total time in sunlight also went up gradually as people were able to tolerate more time. And of course, those of you with EPP are well aware that you have learned to stay out of the sun. So these studies are sort of requiring or asking you to purposely expose yourself to greater sunlight as time goes by to see whether you can tolerate it. In terms of overall quality of life, most patients expressed a feeling of marked improvement, although not all. So there were some subjects who didn't think that they had much of an improvement, but on the whole, patients expressed quite appreciable improvements. And of particular interest, and somewhat surprising, I might say, is that the mean hemoglobin level really didn't change over the entire course. I mean, we would have expected that there would be some decrease in hemoglobin in the earlier studies with Bitopertin, not in people with EPP, but with other disorders. There was a fall in hemoglobin, but that was not really observed. There were some other treatment emergent adverse events, TEAE, that's what that stands for. But they were all grade 1, that means mild in severity, and the most common ones were some dizziness and headache, as you can see here. 60% of people given the active drug described dizziness, but it was not severe and did not require them to stop using the drug. So this was obviously very exciting results for the company as well as for patients. It's really proves the concept that Bitopertin is able to reduce proto overproduction. There was a dose-dependent influence, as you saw on the slides, a significant effect on sunlight tolerance compared to baseline, overall improvement in quality of life, and no meaningful change in hemoglobin. Other safety measures were also really not observed. So no changes in liver tests and kidney tests and cardiac studies, etc. So it seems to be pretty well tolerated, as one would expect. And it's clearly been the basis for going on to additional trials. There has been a trial that we and others have been involved with, and there's now plans underway for a phase 3 trial. There's also a phase 2 NIH-led trial in a condition called Diamond-Blackfan anemia, which is an aplastic anemia, which is expected to start up in mid-2023. So it's obviously early days, limited number of patients, but very exciting results. And I think the other thing is, again, that this would be the first drug that is getting at the underlying pathophysiology of overproduction of protoporphyrin. And unlike either alpha-melanotide SCENESSE, which some of you may have some experience with, or with dersimelagon MT7117, the still experimental orally active drug that increases eumelanin production by interacting with the melanocortin-1 receptor. These are useful, certainly, for improving the amount of pigment and decreasing the severity of photosensitivity, but not getting at the overproduction of protoporphyrin. So Bitopertin is the first drug that looks very promising to do that, and to not only improve the photosensitivity by decreasing the protoporphyrin, but conceivably over time also decreasing the formation of protoporphyrin gallstones, the need for cholecystectomies, and we hope, we're optimistic that it may help to prevent the rare but severe protoporphyric hepatopathy that occurs in a small proportion of patients. So I would say overall it's very exciting results and very encouraging results. And I think Disc Medicine and their team are to be congratulated on these developments.